All right, I'm going to talk now about enzymes and the environment. This is very important information for any student of biology or the natural sciences in general, and it's also really important information for understanding the lactase lab for which you are writing a formal lab report. Enzymes are very much affected by the environment in which they are found. Four particularly important aspects of the environment that impact enzymes very directly are listed here, and they are the concentration of reactants. Remember that reactants also equals substrates. Okay. They are temperature of the environment, pH, and inhibitors. These first three, here, here, and here, are all factors that you tested in your lactase lab. You didn't test the last, but I am going to talk about it in this lecture nonetheless, as it's important information to have. So the concentration of substrates very much impacts the concentration of reactions. In an uncatalyzed reaction, as shown here in blue, as you increase substrate concentration, shown on the horizontal or x-axis, you see a corresponding increase in the rate of product formation, and the rate of product formation, or the y-axis here, is a measure of enzyme activity. How hard those enzymes are working. The harder they work, the more product formed. So in a routine uncatalyzed chemical reaction, as you add more substrates in, as you add more reactants in, you get more product, and it's a linear relationship, and you'll see therefore a straight line as the blue is showing here. In a catalyzed reaction where you have enzymes involved, it's typical to see this profile as shown in red. Okay, where the rate of the reaction increases as you add substrates until you get to a particular level, at which point it plateaus off. This is a profile known as saturation kinetics. And the reason for this is that somewhere about here, or maybe here, okay, somewhere in here, what's happening is all available active sites. And remember, the active sites on an enzyme are those portions of the enzyme that bind specifically to the substrate or substrates. But at some point in an enzymatically catalyzed reaction, as you add more and more substrate, you get to this point where all available active sites are occupied. So even though you add more substrate, you don't get an increased rate of reaction, and you see what's called saturation kinetics. An analogy for this is a factory. A factory is a place where work happens. Here the work is chemical reactions. It could be something else at a factory, of course. The workers in the factory are the enzymes. As you bring more and more raw materials into the factory, in theory for a while the workers can make more and more product, but at some point in time all of the available workers will be working as hard as they can, and even if you keep bringing in more raw materials, you won't be able to get more product out. That's an analogy to explain how the concentration of substrates directly impacts the rate of reactions or the enzyme activity. Another really critical factor that affects enzyme activity is the temperature of the environment. Two profiles are shown in this graph here. Up the vertical axis, or the y-axis again, the y-axis is measuring, it's a measure of enzyme activity. And the x-axis is temperature in degrees C. Okay? So humans would fall in and around here at 37. Okay? So here are two temperature profiles. Let's look at the blue one first. Okay? So what we're seeing here is that as temperature increases, initially the reaction rate increases as well, okay, until you get to a maximum, okay, or an optimal temperature. That is the optimal temperature for this enzyme. Okay? And beyond that, we see a very sharp drop-off. So we'll see a sudden steep decline to a point where essentially we have no activity. And so to explain that profile, you need to remember that all chemical reactions, as temperature goes up, will happen at a faster and faster rate. And that's what's explaining this first part of the curve. Okay? You're seeing more molecular movement, the reactants are encountering one another more frequently, and the reaction is occurring at a more rapid rate. At the optimal temperature, what's happening is the enzyme is in the perfect conformation for the most rapid catalysis of that chemical reaction. And what's happening after that, in this part of the curve on, what's happening there is high temperature is denaturing the enzyme. And what does that mean? The term denature means to lose the 3D structure of a protein due to the breaking of H or hydrogen bonds. So what's happening in these temperature profiles is once you exceed that optimal temperature, okay, you will have such high temperature that in fact it is causing the hydrogen bonds that are holding that protein into its three-dimensional shape to dissociate or to break, and therefore the protein is unfolding, okay? So the protein unfolds. And to get a visual for that, think about what happens if you've got a hot frying pan on the stove and you crack an egg into it, and the egg immediately turns white. The egg goes white immediately, and we think of that as cooking, but what we're seeing is we're seeing the change in the physical properties of the egg protein as it is denatured due to the very high temperature environment. You're just seeing that very, very quickly. So temperature really affects enzyme activity. Enzymes, of course, come from living organisms, and one thing that we know to be true is that the optimal temperature for each enzyme will typically be the ideal temperature for the particular organism from which the enzyme is derived. So this graph shows an enzyme. It's a chitinase enzyme. One is from a bacteria that lives in a very cool environment, and the other from a bacteria that lives in a really hot environment. We'll worry about the neutral and acidic later. So the temperature optimum for this enzyme is much higher when it is derived from the bacteria that comes from a hot environment than the cool environment, and that's because those organisms have evolved to have enzymes that work optimally when at the temperature that the organism is found at. All right, pH or acidity is also an important environmental factor that impacts the activity of enzymes. And pH is a measure of acidity. Therefore, it's a measure of hydronium ions or protons, which are directly related to hydroxide anions. By definition, pH is the negative log of the proton concentration in an environment, and it is expressed on a scale that ranges from 0 to 14. 7 is considered neutral, less than 7 is acidic, and greater than 7 is basic. Because it's a negative log equation that determines pH, low pH equals higher H plus equals more acid. Okay, and high pH equals lower H plus equals more basic. Okay, so these protons and these hydroxide anions are charged, and because they're charged, they're going to be attracted to other chemical species that are positive or negative, or partially positive or partially negative. Therefore, the amount of H plus and OH minus in an environment will impact the hydrogen bonding that can occur. Okay, so pH impacts H bonding. Okay, and it's going to impact the hydrogen bonding that affects substrate attachment to active site and protein folding. So the curves that show the relationship between pH and enzyme activity look a lot like the ones we just looked at for temperature. Typically what you'll see is there'll be a curve like this, where there's some sort of optimum pH at which the protein enzyme is perfectly folded and the substrate is able to optimally hydrogen bond with the active site, thereby promoting chemical reactions. On this side of the curve, we have less optimal active site and substrate binding. And on this part of the curve, what we're often seeing is the protein is denaturing. Okay, down here on the extremes on this side, you can see a little bit of that happening as well. Okay, so pH is an important aspect in driving or determining how well enzymes work. Again, as for temperature, all enzymes come from organisms, and all of those organisms have evolved to have cellular structures and environments that are optimized for the environment in which they're found. So what you'll typically see is that if you take enzymes from organisms such as bacteria that live in really acidic environments, they will have optimal pHs that are relatively acidic. Remember, that's neutral, that's basic, and that's acidic. Okay. Whereas if you take a similar type of enzyme from an organism that lives in a neutral environment, you'll see a pH optimum promoting optimal enzyme activity that is closer to neutral. Okay? If you were to take an organism from a very basic environment, isolate the same enzyme from that organism, and then test its activity, you might see a profile that looked something like this, where the optimum was, quite basic. Now, in your enzyme lab, in your lactase lab, you did not test out the role of inhibitors, but it is important information that you should understand so that you have a good picture of how metabolism happens in organisms on Earth. So we've talked a lot about active sites and how substrates can bind to active sites, and over here we're looking at a green enzyme, and it has 3 pockets, and it's binding to 2 substrates. So really each of these pockets all together comprise the active site. Now, inhibitors, typically we use the word inhibit to mean that we're reducing activity. And there are 2 basic types of inhibitors that can impact enzymes, what are called competitive inhibitors and allosteric inhibitors or regulators. So on the left we have a model showing what competitive inhibition could look like. So competitive inhibition is shown here. And in competitive inhibition, the substrates cannot bind when a regulatory molecule binds to the enzyme active site. So what we're seeing here is, in the active site, this blue molecule is a competitive inhibitor, meaning that it has the same shape and chemistry as the substrate, the natural substrate for that enzyme. Therefore, it will occupy the active site of the enzyme, blocking the natural substrates, shown in orange and red, from accessing the active site. So it's a competition here, right? Both the inhibitor and the substrate compete for the active site. Okay, so if you had a certain amount of competitive inhibitor and a certain amount of substrate, you would have a particular level of enzyme activity. If you added more competitive inhibitor, you'd see the enzyme activity going down. In contrast, if you added more substrate, you would see enzyme activity going out, because it would then be able to outcompete the inhibitor. So the other type of inhibition that is routinely seen in cells is what's called allosteric regulation. And allosteric is derived from a Greek word which means other. And in the case of allostery, what we're talking about are regulatory sites other than the active sites. Okay? So an allosteric site is a regulatory site on a protein. It is not the active site. It's a different site. Okay? So here we see a protein that has an allosteric site, and it is here, and that yellow molecule, that yellow regulatory molecule, is binding to the protein. And when it binds to the protein, it causes a shape change, and that shape change is actually making the active site available, opening the active site up. So you can have allosteric activation, where the active site becomes available to the substrates when a regulatory molecule binds to a different site on the enzyme. In other words, in that case, if there was no regulatory molecule, the active site would not be open or able to receive substrate, and there would be no detectable enzyme activity. You can also have what is called allosteric inhibition, where the regulatory molecule involved binds to the allosteric site, causing a shape change that closes the active site and therefore blocks access of the naturally occurring substrate to the active site. Okay? So when you've got allosteric inhibition, you've got less activity. When you have allosteric activation, you've got more activity when the regulatory molecules are present. Okay, and that concludes our discussion of inhibitors. So as we learn more and more about cells and about metabolism, it's important that you start developing an appreciation for the concept of metabolic pathways. And these are basically connected reactions. They can be pathways or they can be loops, okay, or cycles. And metabolic pathways, this concept is important to address at the same time that you're starting to learn about the idea of inhibition, because a lot of metabolic pathways have a built-in inhibition process that's referred to as feedback loops, okay? Feedback inhibition typically. So let's take a look at these figures and think about how that could be. So here on the left are enzymes 1, 2, and 3. And each of them is catalyzing a chemical reaction, and they're related to one another. Okay? We start with a particular substrate, shown in blue. Enzyme 1 catalyzes the conversion of that substrate to what's called intermediate A, this triangle thing. Okay? That is the substrate for enzyme 2. So it binds to the active site in enzyme 2, and enzyme 2 catalyzes the conversion of intermediate A into intermediate B. And this little kind of kite-shaped thing is supposed to be intermediate B. Intermediate B is the substrate for enzyme 3. So it'll bind to its active site, and that will catalyze the formation of the final product of interest. Okay, this is something that the cell needs, presumably. So we start off with this initial substrate, and we subject it to 1, 2, 3 reactions in order to get a final product. And that's what I mean by being interconnected. The product of one reaction is the substrate for the next. Now note this feedback inhibition arrow. What that's showing us is that the final product of the pathway is actually acting at a site other than the active site for enzyme 1, so that would be an allosteric site. And when that product binds to the allosteric site, it results in what is called feedback inhibition. So this is a kind of allosteric inhibition. We use the term feedback because we're getting the regulatory molecule is actually the product from a multi-step biochemical pathway. So this really makes a lot of sense to do things this way, all right? Because in this, it allows the cell to control the level of product, of the final product, in the cellular environment. So when there's a lot of this final product, then a lot of it will be available to diffuse over to the allosteric site on enzyme 1 and to bind to it. And when it does that, it shuts down this active site so that that substrate can no longer bind, and then that enzyme is no longer generating intermediate A, so enzyme 2 isn't working. Enzyme 2 is not generating intermediate B, so enzyme 3 can't work, and if enzyme 3 can't work, then you're not continuing to make more of the product. So this is really useful for maintaining specific levels of a final product in cells. Okay? And it also allows cells to conserve energy, particularly in the case of anabolic pathways. All right, so with that, I'm going to conclude this mini lecture.